So the topic which I chose to look at is, I chose to look at mitochondrial DNA. Now I know this doesn't really seem like a specific topic that's similar to other people's topics in the class, because other people tend to present topics which are about one mutation or one anomaly or one disorder. But mitochondrial DNA and mitochondrial genetic anomalies, the severity and the symptoms and the presentation really does tend to run the gamut of not detectable at all to severe and lethal. Um, mitochondrial genetic anomalies really kind of hit close to home for me. I'll explain why just a little bit later. But I think it's very, very important that we understand how mitochondrial DNA works, how the inheritance patterns, and the big differences between mitochondrial DNA and nuclear DNA. So, the mitochondria was not always a part of the cell as we know it today. To consider when the mitochondria joined the cell as we know it today that became the eukaryotic cell, you kind of have to accept a couple of assumptions, and there's a lot of research and there's a lot of different opinions that are out there based on this. But the one that I tend to go with is the theory that the Earth was created about 4.5 billion years ago. Based on this particular theory, it can be seen that there was really not any life as we know it for about 1 billion years with a B. We're talking long periods of time. So for that first billion years, not really much was around. And then at about 1 billion years, because of a collection of this and a collection of that, prokaryotic cells or bacteria really began to dominate the Earth. This continued for a bit, about another 1 million years, when because of the prokaryotic cell and bacteria's presence and the beginning presence of chloroplasts, which are associated with with oxygen production, which is a whole separate issue and a whole separate presentation in itself. About 2.5 billion years ago, the oxygen accumulation in the atmosphere began to increase. This oxygen accumulation in the atmosphere really didn't have any direct effects on cellular life as of then, but as things progressed throughout another 1 billion years, bacteria began to use oxygen through an aerobic process. So what I want you to picture is about 1.5 billion years ago, a primitive cell was kind of hanging out. It may have been, may have just left the the prokaryotic cell bar. And it's like, hey, I want to get some water through some pinocytosis. And then, it drank too much, and, that's what happens. And then you make decisions that you always regret. In this particular case, it was one of those decisions that turned out to be pretty positive. So what happened was, this primitive cell went to go and ingest some nutrients through phagocytosis. So through phagocytosis, it took in a bacterial cell. It was planning to, digest this and eat it. Well, after it took in the bacterial cell, just like normal, a vesicular membrane was formed around it, and this vesicular membrane is normally what would have fused with the lysosome, and the contents of it would have been digested. But what happened is the glucose, which was present in the cytoplasm, was able to be diffused through both membranes and allowed to access the mitochondrial cell, or the bacterial cell. It obviously wasn't a mitochondria back then. So as this happened, the mitochondria began to produce ATP, and it always produced ATP, but the ATP which it produced was then able to power the metabolic processes of the cell. Now, these metabolic processes were able to function a lot more efficiently with the large amount of ATP that was produced compared to the non-oxidative phosphorylation which was occurring before, or the substrate level phosphorylation as it's referred. So, the cell did not lyse the contents of that particular vesicle, and prokaryotic cells, being their own independent structure, they stayed with us, they divide, and we'll get into exactly why that happens right now. So, the organelle structure for mitochondria, as you can see here, the mitochondria is a basic prokaryotic cell. The contents of it, you can see the little small speckles. These small speckles are ribosomes. You can see circles here. These circles are the genetic information. We'll get into specifically how the genetic information differs from nuclear DNA here in just a minute. And then you can see proteins floating around. So you have a big endoplasmic reticulum, you have the proteins, you have the DNA, and then you have the outer, let's see it here, the outer membrane, and this outer membrane is really what plays a major role in ATP production. And then you have, I'm sorry, that's the inner membrane, and then the outer membrane here is the membrane which had wrapped around the initial bacterial cell, which was uptaken through phagocytosis. So this outer membrane here is associated with the vesicle that was created. You can see a transmission electron microscope view here. You can see a few ribosomes that are speckled, and you can further see the matrix as it goes across the cell. The reason the matrix goes across the cell is because that increases the surface area and makes it more efficient in its function. So mitochondrial DNA, as they once were prokaryotic cells, they do reproduce based on their own clocks, their own schedules. They also there tends to be a lot of them in one particular cell. So how we can consider this is it's its own entity. It's going to divide at its own rate, and when it does divide, it's going to divide via binary fission. Now, binary fission is a very important concept to grasp, which I'm not really going to go over in great detail, but it comes into play in the next cell. You can easily see here in this particular picture the number of mitochondria which are present in a cell. Now, the particular number that are present vary anywhere between 100 and 1,000 per cell, and it's a little difficult concept to grasp that one cell can have 1,000 of something else in it, especially considering the fact that cells, we normally consider them to be so small. But here you can see that as the phospholipid bilayer present in the mitochondria have been stained green, you can see how many of them are present in the cytoplasm. They're all extranucleus, so they're all outside the nucleus of the cell. The blue would be the nucleus, and the red here would be the small microtubules which are present to keep the cell in its original shape. I guess it doesn't get all ripped apart by shear forces and big cellular bullies and things like that. So as we continue, you have to consider that the mitochondrial DNA and the nuclear DNA, they vary greatly. A couple of ways that they vary, the most obvious one you can see here. Now, I included a copy of the mitochondrial DNA, and this is a full copy of all the proteins that are on the mitochondrial DNA. It codes for about 37 genes, and then obviously it would be a little bit difficult for me to upload the entire human genome into this particular presentation. I just included kind of a snippet of one gene. So when we look at this, you can consider that mitochondrial DNA contains codes for about 37 genes, whereas nuclear DNA contains codes for anywhere between 21 and 22,000. Now, as kind of a side note, when the Human Genome Project finished, it was really a hit to everybody's ego, because originally it was thought that nuclear DNA coded for about 100,000 proteins. And everybody's like, oh, we're awesome, and then, oh, we're not quite as awesome as we thought. But we're still pretty cool, so that works out. When the aliens come, we have something to brag about. There's still a lot of genes. Anywho, so mitochondrial DNA tends to be circular, as you can see here, which is similar to prokaryotic DNA. And nuclear DNA is linear. Mitochondrial DNA is obviously a great deal shorter than nuclear DNA, whereas mitochondrial DNA contains about 16,500 base pairs, whereas nuclear DNA contains about 3 billion with a B. Now, I don't know about you, but I don't want to count every single one of those. That's why we have magical computers, and, short people gotta have jobs too. So, anywho, let's continue. Mitochondrial DNA is only inherited by the mother, or through the mother's bloodline. Now, I know what you're thinking. You're like, hey, that doesn't make any sense. Because, we went through and learned about Mendel, and Mendel was talking about haploid and diploid and, things like that. But, so why would one set of DNA not go through crossing over in prophase and things like that. And to understand that, you really have to remember that the mitochondria were initially a prokaryotic cell. With them initially being a prokaryotic cell, you have to consider that mitochondria replicate via binary fission, not via mitosis. So there was normally no crossing over. So essentially, one mitochondria is a clone of every other mitochondria. There's going to be small changes in the genetic code, which really does come into play as this presentation continues. But you got to understand that's the reason that mitochondrial DNA in the cell is only present from the mother. So why is this, even though mitochondria are present in the sperm? Well, there's a couple reasons. One is the sperm contains anywhere between 100 and 1,000 mitochondria, whereas the egg contains anywhere between 100,000 and 1,000,000 mitochondria. So just based on the simple facts or the simple numbers, the presence of the egg mitochondria would just extremely dilute the presence of the sperm mitochondria. Also, with this, the mitochondria don't easily pass into the eggs, don't easily pass through the egg cell membrane, thus keeping most of the mitochondria within the sperm or within the extracellular space. And also, paternal mitochondria are rapidly degraded by the presence of autosomes that are present in the egg cytoplasm, thus perpetuating the fact that the mitochondria that are present are going to be from the mother. So, why would we focus on mitochondrial DNA? It seems like, the mitochondria have been around for a long time. Why would we really take the time to focus on this particular little tiny, tiny organelle that you can fit thousands of in a normal cell? Why would we focus on this? And the reason that we would focus on this is because of every single one of these major disorders. Now, some of these disorders here, as you can see, they're not super serious. They're not really life-threatening. We can treat diabetes now. It's not a real big deal to have short stature. Hypothyroidism, we can treat. But a lot of these particular anomalies are lethal and severe, and they result in failed pregnancies. And based on the inheritance patterns, you really have to consider that this is a great effect for families. So with this, you have to consider that since the mitochondria, their purpose, the reason they were saved inside the eukaryotic cells is because of their ability to produce ATP. So cells that have more mitochondria is the cells that are more energy hungry. So muscular cells tend to have more mitochondria. Neural cells tend to have more mitochondria. Cells that are kind of sitting there and hanging out, epidermal cells, skin cells, they don't tend to have as many mitochondria. So you see here, you don't tend to get anything like psoriasis listed down here. You tend to get things that are pertaining to muscular function and high neuro conditions, such as seizure, that's a neuro issue. Myoclonus, which is a muscular issue. Blindness, migraine issues, which is associated with neural function. Dystonia, which is, decreased muscle function. Peripheral neuropathy, that's a peripheral nervous issue. Weakness, blindness, cataracts. Diabetes, like I said, we can tend to treat that. But a lot of these things are very, very severe. Lactic acidosis, especially if it occurs in infants, causes irreversible neurological damage. And there is some research that shows that once the lactic acid level reaches a certain point, it causes irreversible, like near 100% morbidity for infant and pediatric patients. So that's the reason that assessing mitochondrial disorders and seeing their presence and us developing a way to ultimately treat them or fix them it really should be the goal. Now, mitochondrial genetic disorders are a very, very common, relatively, cause of severe defects immediately after birth. Now, the numbers that I'm getting ready to give you only include severe defects at birth. But the numbers are a lot higher if you consider mitochondrial diseases that come up as the person's lifespan continues, but these tend to be the most severe ones. So about 4,000 U.S. births annually tend to have severe mitochondrial disorders, and the reason these are all about is because there's not data out there for every single kid that's born. We don't assess every single child for this. And so even if a child has something that could be associated with mitochondrial disorders, we don't always assess the mitochondrial DNA, primarily because by that point, you can assess it, 7 days a week and twice on Sunday, but you're still not going to be able to change anything. You can't treat it at that point, which is really the problem that we have. So the severity of the orders are the disorders really vary based on when the particular anomaly occurs, but a lot of the time they're degenerative, particularly if they occur in infants and young children. Also, due to inheritance, they are often repetitive and they affect multiple, if not all, children. So, when is mitochondrial DNA mutated? This is perhaps the most important question of anything else in this presentation, and understanding when the mitochondrial DNA is mutated really gives you a better idea of, first of all, how severe the mutation is going to be, and second of all, how many cells or what percentage of each cell is going to be affected. There's 4 primary types of mitochondrial genetic mutations. They include maternal primordial oocyte mutations, and that would be when the mother is still a fetus. So the mother, back a long time ago, before they hit puberty, before they were born, when the eggs were initially formed, the initial stem cell that created all of her eggs had a mutation at that level. The next one is a single or multi-follicular mutation, and that would be eggs were formed. Those eggs developed into follicles, but as those follicles developed, then there was a mitochondrial anomaly that occurred there. There's germline mutations, and germline mutations occur primarily as the fetus begins to develop and through cellular mitosis. And they can also develop later in life and later in childhood or adulthood, but I'm really not going to get into that much here because you really can't treat it with what I'm looking at. So the first one we were looking at is primordial oocyte mutations, and if you consider that before birth, there's the oogonium, which is a stem cell. This stem cell is the one cell that's going to undergo mitosis and mitosis and mitosis lots and lots and lots of times, and it's going to copy itself. And when it copies itself, this is what's going to become all of the eggs that are present within the woman's ovaries throughout her life. So as we consider this, if there is an issue with this particular stem cell, if there's a mitochondrial genetic issue with this particular stem cell, when the mitochondria undergoes, or when the oogonium undergoes mitosis, and the contents of the cytoplasm are split, if those mitochondria are anomalous, or if they have issues, every single egg is going to have the same issues because there's no crossing over. The genetic material in the mitochondria is the exact same from the stem cell through every single follicle that's produced, every single oocyte that's produced. Now, the oogonium tends to undergo mitosis anywhere between 5 and 30 weeks gestation. Remember, a normal fetal gestation is about 40 weeks. And in this particular type of anomaly, where it's the primordial oocyte that's affected, all of the oocytes that are produced, all of the follicles that are produced, are going to be affected. So in this particular case, every single child that this mother has is going to be affected with whatever mitochondrial disorder is associated with the anomaly. She can try as often as she wants, but every single egg is going to be affected. And this is one of the primary types of mutations, which does happen very, very often, especially today. And it tends to be the most devastating for families. The next thing is a single or a multifollicular mutation. And I really like this picture. I've used it a couple times in this particular presentation. And consider that if you have the primordial oocyte over here, the primordial oocyte undergoes mitosis. Now you have two. Each one of those undergo mitosis. Now you have four of them. And then consider that there is an issue with the mitochondrial DNA at this level. Then, since you're at the level of four oocytes present, that means that 1/4 of all of the mother's eggs are going to be affected with this mitochondrial disorder. And consider the exponential growth factor, and the hundreds of thousands of eggs that are present inside the ovary. 1/4 of them, or effectively 1/4 of this woman's children, are going to be affected. Furthermore, the latter down the germline this particular issue occurs, the fewer of the eggs are going to be affected. But the reason I really like this particular issue, the reason I like this particular diagram is because it also shows that if you have the primordial oocyte, and remember that within each one of these you have anywhere between 100,000 and 1 million mitochondria. So consider this. Consider that you get down to this point here, and when this particular oocyte undergoes mitosis, let's say a small number, let's say 10% of the mitochondria that are present in this particular oocyte have some sort of genetic mutation. And it's the same genetic mutation for all of them, so it's easy to understand. But consider that 10% of the mitochondria here are going to be affected, 10% here, 10% here, because they go through, the mitochondria undergoes binary fission. So in this particular case, you're going to get an affected patient, but it's not going to be as severe as if it's every single mitochondria. This is where you get the variability in symptoms and the strong variability in severity of symptoms. So I know this is a little bit of an abstract concept, but I have faith you can do it. So, following this, there are germline mutations, and germline mutations are really something that we're also not going to look at a lot in this presentation, but we'll look at it, just so you can kind of get an idea. So germline mutations, we go back, the follicle was okay. This never happened. We went down one of these particular paths, and now we have a normal zygote. The zygote is fertilized, everything's happy, yay, we have happy sperm, we have a happy egg, and then it undergoes mitosis. When it undergoes mitosis, the mitochondrial DNA in one of these mitotic events undergoes some sort of mutation. Now, this is more severe the higher up the germline it goes, but consider in this particular example again, when you're really small, like 4 cells small. This is back when you really got to go shopping at the Baby Gap. Consider that the higher this occurs, the more severe the symptoms are going to be for that particular mitochondrial disease. So as we continue, you can see, obviously the lower it is, the smaller percentage of the cells in your body are affected. The higher it is, the more number of cells. You also have to look at the number of mitochondria per cell that are affected and understand that the mitochondria are going to be fairly equally divided during cytokinesis in the process of mitosis. So, the incidence of severe deleterious mutations are more likely in mitochondrial DNA compared to nuclear DNA. Now, how often is this? Some research has shown that it's about 10 times more likely in mitochondrial DNA compared to nuclear DNA. Now, why is this? Well, prokaryotic cells, while they do have DNA repair mechanisms, they're not as complex as eukaryotic cells. And this actually has changed a bit in the past 5 or 6 years, because in the past it was believed that some prokaryotic cells lacked any ability to repair their own DNA. Now we know that they have some. There's the SOS repair mechanisms for UV light and things like that. But despite all of this, the mitochondrial DNA anomalies tend to encompass about, they tend to occur about 10 times more likely than in nuclear cells. What also makes it relevant is since there's so many of them per cell, especially in early development when mitosis is very active in most of the cells, neural cells, muscular cells, things like that. Um, these are that you're going to have a lot more divisions than you're going to have nuclear divisions. Also, since non-mitotic cells or cells in the G0 state, the mitochondria still divide. The mitochondria are still, degraded. New mitochondria are produced. So people run the possibility of developing a mitochondrial genetic mutation, even in G0 cells that are not undergoing mitosis. And really, to understand how serious this is, and what the bigger differences are, and how often this can occur, you have to consider the functional portions of the mitochondrial DNA. And really, we understand the nuclear DNA a lot better, so we have to consider those both put together. So we already kind of discussed this, but there's one more kind of spin that I want to throw on it. And that spin is, first of all, what's different about nuclear DNA and mitochondrial DNA? I'll help you cheat. Here are the pictures that we had earlier. You can see that mitochondrial DNA is circular and nuclear DNA is linear. Okay, that's cool. We also know that mitochondrial DNA is a lot shorter, about 16,000 base pairs, and nuclear DNA is longer, maybe 3 billion base pairs. We also know that the number of genes that are encoded on mitochondrial DNA is about 37, and on the nuclear DNA is anywhere between 21 and 22,000. But so we have all that information here. But is there anything else that you see different? I'm going to pause here for just a second and let you be like, wahaha. So what I'm trying to get you to consider is if you look on the linear genetic segment here, we have this portion right here between the enhancer elements. We have this portion right here between the enhancer and the promoter. We have the section here between the promoter and the Tata box. All of these are untranslated regions, or UTRs. We also have introns that are taken out and thrown out to the cats. We have more untranslated regions and some untranslated regions that are going on between the other enhancer elements. And if you look up here at the mitochondrial DNA, this picture doesn't really show any genetic material that doesn't pertain directly to protein synthesis. Now, that's really not entirely true. And the reason I say that is because you have to consider how much of the DNA that you have is functional. So I threw a fourth little variable on here, and I didn't really have this one in the earlier one, but as the mitochondrial DNA contains about 16,500 base pairs, and this is really true of most prokaryotic cells, only about 2% of the DNA is considered junk DNA, and that would be DNA that doesn't directly code for proteins. So there's 331 nucleotides that are present in prokaryotic DNA that don't directly code for proteins. In our nuclear DNA, it is 98% of the human genome does not directly code for proteins. Now, if you just compare the raw numbers, in mitochondrial DNA there are 331 nucleotides that directly don't code for proteins. In nuclear DNA, you have 294 million. Okay? So, and I added 3 extra zeros, so you got to forgive me here. No, that's right. 294 million nucleotides that don't directly code for proteins. Now, there is some research in both prokaryotic cells and eukaryotic cells that says that, a percentage, up to 40% of that, assists with expression of other genes, but there's really kind of up in the air. So even if you just look at the raw numbers, you could have a mistake in the DNA in one of 294 million different spots, and you're not going to have a lot of noticeable effects. However, in the mitochondria, that can only occur in one of 331 spots for you not to have noticeable deleterious effects. So now you're beginning to see why mitochondrial genetic anomalies are so much more likely. So even if the mitochondria possessed some magical super ability to repair every genetic mutation, it would have to be about 100 million times more effective than nuclear DNA for you to get the same number of anomalies between the two. Hence the issue. So, if patients are presented, or if patients do present with severe mitochondrial genetic anomalies, what can we do for them? Well, the answer to that is the same thing that's really been mentioned in a couple of presentations, and that would be that supportive care is the primary treatment. Now, what does supportive care encompass? And I think this is really important to understand. Now, think back for yourself. Have you ever had a disorder or a disease or an illness where you go to the doctor and they just prescribe you supportive care? And I know a lot of you are going, nope, that's not me. I'm very healthy. Yay, I'm so young. But if you ever gone to the doctor and had a cold is something that's commonly provided for supportive care. Now, what is supportive care? Pretty much nothing. So we treat symptoms, we treat things as they come up, but we're doing nothing to treat the underlying physiology. An example of this, in my opinion, is, we have all this technology out here, but when someone goes blind, what do? Not that I don't love dogs, but we hand them a dog and say, here, let the dog drag you around for a while. That'll take care of it. If we have someone that's paralyzed, we say, oh, you can't walk? Can you sit? Oh, okay, cool, we'll give you a chair. You can do that. And this, this concept of not being able to do everything makes people very sad. So I don't really know who this woman is, but she was wearing oxygen, and, it always just relates to me. So there you go. But what if? What if you had a particular mitochondrial anomaly? I'm assuming you're pretending you're a woman right now. You had a particular mitochondrial anomaly that occurred at the primordial oocyte, so every one of your eggs was going to be affected by some sort of severe genetic mutation that was going to result in every one of your children being wheelchair-bound and only living for a short period of time. Now stop and think about that for just a second. Now, you might just say, oh, well, I'll just adopt. Okay, and that'd be super. That would be a solution to your issue. Congratulations. But consider that adoption is not for everyone. While I think it should be, it's not for everyone. And also consider, a lot of you guys know that I worked in the Middle East for a long time. In the Middle East, having kids is not an option. It's expected by the families, and they're not only expected to have kids, they're expected to have a lot of kids. It actually goes so far as to be a tenet of Islam. Go forth and multiply, I believe is what they say, and believe me, they go with that. So, what if you had this severe anomaly, this severe mutation, but somebody were to come up to you and say, hey, what if you could decrease the chance that your child is going to have that mitochondrial disorder to the exact same as a normal person? What would you do then? Well, most of you would be like, dude, I'd totally go for it. So that's what we're going to look at now. Now, that brings us to nuclear transplantation. And what is nuclear transplantation? That's not picking up a nuclear power plant and bringing it somewhere else. No, no, no. That would be transplantation of a maternal nucleus into a donor-enucleated primary oocyte or a donor-enucleated primary egg. So essentially, you're taking the nucleus of the mother from the egg that has the bad mitochondria, and you're putting it into another egg where you've taken the nucleus out of it. So, the eggs are harvested from the would-be mother. The eggs are harvested from the egg donor. Now, how does this happen? Both of them are given a human gonadotropin, which stimulates the maturation of the follicles. After the follicles are stimulated, they're given human chorionic gonadotropin, which stimulates the release of these mature oocytes into the fallopian tubes and down into the vagina, or into the superior portion of the cervix. Sorry. These eggs are then harvested via a transvaginal needle aspiration. Sorry. And the eggs are saved outside or in vitro. Now, how is the sperm harvested? Well, it's generally taken from the father and involves a dirty magazine and some private time. You can use your own assessment as to what happens in that private time, but we're just not going to go into that here. So, essentially, as you can see in this video over here, it's not a super complicated process to enucleate a cell, and that's actually the video that we have playing over here. So what happens is the donor and the maternal oocyte are treated with an enzyme PI3K, and this particular enzyme is reversible, and it arrests the cell cycle at the SG2 checkpoint. So when this happens, the DNA is a little bit bigger because it's a little bit spooled out, so it's easier to see underneath a microscope. And the cells, the eggs, are put under a microscope, as you can see. This is a small pipette which produces a suction which holds the egg in place. This is a hair-thin needle, and as you can see in this particular example, they secure the egg. The hair-thin needle is pushed through the membrane, and this is the genetic material here. It's sucked up with a small portion of the cytoplasm, and it's ejected outside the cell. So the cells that as they leave, they have no genetic material, with the exception of the genetic material that's present in the cytoplasm, which would be within the mitochondria. So after this is done to all the cells in question, and this would be an egg, so this is before it's fertilized, a maternal nucleus, through the exact reverse process, is placed within the egg, and these are the nuclei that were removed from the mother's egg in this exact same fashion. And they are, as a general rule, they just kind of sit there. Then what happens is they take the father's sperm and they fertilize the egg. Now there's two different ways to do this, as you can see from both of these really cool little videos down here. The first way over here is via direct exposure, and that would be you have an egg. This is a newly nucleated egg. With it being a newly nucleated egg, they take anywhere between 50,000 and 100,000 sperm, and they essentially pour it on top. The sperm do their job, and the sperm will attack the egg. When they attack the egg, only one sperm is allowed in. And this works if the sperm is motile. But if the sperm is immotile, what happens is they have to go through and manually inject a single sperm into the cytoplasm of that egg. Now that seems like it'd be a lot more complicated, but it's seen in this particular video here. We have the same ultrafine needle. The ultrafine needle pierces the membrane of the cell, goes the external membrane and now the internal membrane. It's going to push through. As it pushes through, you can actually see the sperm within the needle here. It's not in the noodle. They have to be very, very careful to not push through the opposite side of the cell. And then the genetic material is injected into the cytoplasm, and the needle is removed. Now, you seem like, oh, we're all done. Well, we're not so much done, because then you have to look at the cell and make sure that you didn't mess up any of the genetic material inside the nucleus or inside the mitochondria during this particular procedure. So the embryo is screened, which is actually very, very similar to the 23andMe website. The embryo is screened for all known genetic diseases, including genetic diseases which can involve adulthood, Huntington's disease, cystic fibrosis. There's at last count, according to a few of the sites that I looked at, there are about 6,000 inherited diseases which are screened for by this particular pre-implantation genetic screening. After the genetic screening is completed, the blastocyst embryo, which you can see here, which is kind of a ball of cells, is implanted into the mother, and hopefully, she won't have this kind of like alien thing going on right here. But if she does, I'm pretty sure she'd be happy, because that means there's a kid in there. So, the genetic implications of doing this. Now, this is what I think it gets really cool, is because you have to consider the DNA of the progeny. The DNA of the progeny, we've always learned that, kids or people are diploid, and nuclear DNA, and, father and mother, and, recessive, and, whatever. So the child's DNA will be diploid, just like normal. Some will come from the mother, some will come from the father, and that's cool. But the child's mitochondrial DNA will exist from the egg donor. And since the mitochondrial DNA will exist from the egg donor, the child will, depending on your perspective, have three parents: two mothers, one nuclear mother, one mitochondrial mother, and one father. This in itself raises a couple of other issues, which I'm not really going to get into here, but, legal issues have come up. Um, that's kind of for a different area, but I'll get into it in just a second for maybe a minute. So, limitations to technology as it exists today. First of all, the big thing is legality. In a lot of locations, the legality is in question. As of this moment, this particular procedure, it is illegal in Canada, and it is illegal in England. But there is currently an attempt to repeal it in the UK. It's currently in the Parliament, and it looks pretty good as to that it's going to pass. Canada traditionally has been very, very conservative with regards to any sort of genetic manipulation. Canada even makes it illegal to perform pre-implantation genetic screening and to choose the sex of a fetus, even if the parents both carry a lethal sex-linked anomaly. So Canada tends to be very, very conservative in that regard. Here in the U.S., it is illegal in 13 U.S. states, and in almost all of the other states, it's very, very unregulated. It remains unregulated because it's not considered cloning, so that would be illegal in just about every state. But it's not just pre-implantation genetic screening and in vitro fertilization either. So in many states, because this is so new, it tends to be very, very regulated. There's a lot of ethical and religious concerns with it because we're not so much treating a disorder as we are eliminating it. We're affecting the person's genome. Whether it's the nuclear genome or not, we're still affecting the genome. And then the final thing, which is a big major factor, is it is hugely cost prohibitive. That would be the cost of in vitro fertilization by itself for an infertile couple is anywhere between $12,000 and $14,000. The pre-implantation genetic screening, which must be conducted with a nuclear transplant, adds about another $6,000 to the cost. And then nuclear transplant, just the cost of enucleating a cell, renucleating another cell with the original genetic material, and the cost to acquire those cells is anywhere between $10,000 and $12,000, bringing the overall procedural cost anywhere between $30,000 and $40,000 out of pocket. And consider that this is not always successful the first go around. So many people wind up having to do this 2 and 3 times. So they'll spend $90,000 to $120,000 if they have to do this multiple times. Now, there are not a lot of centers that are doing nuclear transplantation at this time, but the number of centers is increasing. The technology is in itself has been out there for about a year, but some of the research that I've cited, as you can see in the references at the end, is as recent as 4 and 6 weeks ago. So this is still very, very new. But there is a lot of promise, and what I think is more than promise is it's actually being used to not just say, oh, well, theoretically in the future we can go through and help us to decrease the incidence of something. It's saying right now we can stop the proliferation of this particular type of genetic issue, and we can make it so families that otherwise have chosen to not conceive because they don't want to have suffering of their children because of these severe, unavoidable mitochondrial genetic mutations, now they can have kids and their kids can have kids. So it makes it so essentially their family can proliferate like normal. So, that being said, I would normally ask you if there are any questions, but you're not here with me, so you can ask all day. I won't hear them. So go ahead. All right, that's cool. So thank you for your time. I know it was a bit fast, but thank you very, very much. Here is a couple of the references that we have. There's a lot of references. It tends to be it's a very modern topic. A lot of people are working on it, um, but all the sources are fairly new. Um, you kind of get the idea. So mitochondrial DNA, I really feel that assessment of it is a major player in the future of genetic diseases and the future of avoiding genetic diseases. Not super future, but like today, tomorrow. So thank you very much, and you have a good day.